Discover how a single gene can produce multiple different proteins through alternative splicing. Alternative splicing is a remarkable process where a single gene can be read in different ways to produce multiple distinct proteins. First, let's understand the basic structure of a eukaryotic gene. Eukaryotic genes contain two main types of sequences. Genes are made of exons, which code for proteins, and introns, which are non-coding sequences between exons. Exons are the coding sequences that remain in the final mature RNA. Introns are non-coding sequences that get removed during RNA processing. Let's see how normal splicing works before exploring alternative splicing. During normal splicing, the introns are removed and exons are joined together to form mature messenger RNA. A large molecular machine called the spliceosome removes the introns and precisely joins the exons together. Now let's explore alternative splicing and how it creates diversity. Alternative splicing allows different combinations of exons to be included or excluded from the final RNA. The same gene with 5 exons can be spliced in different ways to create protein A using exons 1, 2, and 3, or protein B using exons 3, 4, and 5. There are several distinct types of alternative splicing mechanisms. Exon skipping is the most common type, where an entire exon can be included or completely skipped. In exon skipping, the middle exon can either be included to create one protein variant or skipped entirely to create a different variant. In mutually exclusive exons, only one exon from a pair can be included at a time. Alternative splice sites use different boundaries for the same exon, making it longer or shorter. Let's understand the molecular machinery that controls alternative splicing. The spliceosome is a massive molecular machine composed of multiple protein and RNA components that catalyzes the splicing reaction. Splicing factors are proteins that bind to specific regulatory sequences on the RNA to either promote or inhibit the inclusion of particular exons. SR proteins are splicing activators that enhance exon inclusion. H and RNP proteins typically act as repressors that promote exon skipping. Alternative splicing is tightly regulated in cells. Alternative splicing is regulated by tissue type, developmental stage, and environmental signals, allowing cells to produce the right proteins at the right time. Different tissues in the body use distinct splicing patterns to create specialized protein variants suited to their functions. Let's look at a real example of alternative splicing in action. The DSCAM gene in fruit flies can theoretically produce over 38,000 different protein variants through alternative splicing. A single DSCAM gene with multiple exon clusters can be mixed and matched to create thousands of unique protein variants for neural development. Alternative splicing has profound biological significance. The human genome has only about 20,000 genes, but can produce over 100,000 different proteins through alternative splicing. Alternative splicing dramatically increases protein diversity without requiring a larger genome. It enables different tissues to create specialized protein variants optimized for their specific functions. Alternative splicing provides evolutionary flexibility, allowing organisms to adapt without changing their DNA sequence. Defects in alternative splicing can cause disease. Mutations that disrupt splicing can lead to various genetic diseases. About 15% of all disease-causing mutations affect splicing. Cancer cells frequently exhibit abnormal alternative splicing patterns that promote tumor growth. Understanding alternative splicing has opened new therapeutic strategies for treating genetic diseases and cancer. Let's summarize how alternative splicing creates protein diversity. A single gene can be read in multiple ways to produce many different protein variants. Exons can be included, excluded, shortened, or lengthened to create different combinations. The process is carefully regulated by splicing factors responding to cellular and environmental signals. This elegant mechanism enables tremendous protein diversity from a relatively small genome. Alternative splicing is critical for normal development, and its disruption can lead to various diseases.